Welcome everyone to the 2022 edition of the Innovation of the Year here at University of Guelph. Since 2016, we've been recognizing fantastic researchers at the University of Guelph who have converted their discoveries to innovations that have impact in everyday people's lives. Great successes in this regard by a wide variety of researchers. It gives me the greatest of pleasure to be here today with one of the recipients of the 2022 Innovation of the Year Award, Professor Robert Hanner. Bob, great to have you here with me today.
Thanks, Malcolm. Great to be here.
And so, Bob, your particular innovation is one that actually is built, one might say, on the shoulder of giants, you being amongst those giants. Great work in using DNA in an environmental context to understand the world around us and to really make a difference in that understanding in a way that benefits the world at large. Tell me a little bit about that innovation. More specifically, do a better job than I did in terms of defining the specifics of that and how it actually works to make an impact.
Well, we'll start with your comment about standing on the shoulders of giants. And, here at the University of Guelph, we are the global headquarters of the Barcode of Life initiative. So colleagues like Polly Bear and many other researchers around the world have been using short, standardized DNA sequences as a tool to identify species. And that's really powerful for a broad diversity of applications because species identification can be very hard, but it can have a lot of socioeconomic and even legal implications. So as we build these big DNA reference sequence libraries that we could use to identify unknown remains, we showed that there was a lot of fraud in seafood and other commodities. But what we found was that sending samples in for DNA sequencing took a fair bit of time, and there was a cost associated with that. Yeah. And so for a lot of species identification needs, you'd love to have something that's more rapid, potentially even on-site in real time, to answer questions of relevance to industry. So we used a technique called quantitative PCR, where instead of sequencing a whole gene of a barcode marker for a sample, we could exploit the species-specific patterns of variation in its DNA to make a molecular probe that would bind only to that species. So we could then do a quick PCR test and get a red light, green light answer. Either it is the species you're interested in or not. And that proved to be pretty powerful to take to point of need. And we demonstrated that in food and some other commodities, including work with Agriculture and Agri-Foods Canada, Canadian Food Inspection Agency, where you have potential quarantine pests coming across borders. So we started to develop qPCR assays with a PhD student, Amanda Naum, in my lab, to identify aphids of quarantine significance that would be very difficult to identify morphologically. So then, fast forward a few years where people began to realize that we could extract DNA from a water sample and sequence that sample and start to get a readout of the community of fishes or other species in that sample. So using what we call environmental DNA. We're all flaky, right? We shed cellular debris into our environment through a variety.
Around the bathtub, as an example.
So through a variety of processes, and so modern molecular techniques turn out to be so sensitive that we can start to perform biosurveillance in a passive way of just sampling water.
Oh, really cool. So, I think a lot of people are familiar with PCR now because they've used a PCR test to determine whether they have SARS-CoV-2, the virus that causes COVID-19. So what you're doing then is equivalent to that, as opposed to doing a swab of the inside of your nasal cavity and then taking that sample and doing a PCR reaction to determine whether there's a virus there. You're doing the same thing where you, rather than swabbing, you're taking a sample of water, for example. You're doing the equivalent of swabbing the ring around the bathtub. And then on the basis of that sample, do that same polymerase chain reaction, that PCR, to determine the DNA that's there of given species, then to determine whether they're an invasive species or whether there's something that's rare in the environment. Or whether, in the case of the food fraud work that you've done, the food in question is the food that you think that it should be.
That's right.
And so all of those are examples then of environmental DNA sort of detection?
Yeah, potentially. I mean, I would say the food fraud work was more of a kind of a forensic investigation. But what we began to realize where there was commercialization opportunity is that around all of our large-scale development projects, there are laws requiring baseline biomonitoring, ongoing environmental effects or impact assessments. So there is a large industry of environmental consulting that works with primary industry to conduct these kinds of surveys. So looking for fish, they would normally use electrofishing, netting. And so when we're talking about species at risk, our conventional monitoring approaches can actually cause stress or even mortality on the very things we're looking to protect.
Right.
So with the precision biomonitoring approach to commercializing some of these tools, our objective was really to develop qPCR assays for species at risk, so that we can do a better job of understanding habitat occupancy. If we want to protect something, we need to know where it lives. And that isn't always easy to do with rare species.
Right. And are you guys doing that on site then, or are you sampling on site and then doing the testing in a laboratory environment?
Great question. So we had partnered with another company that developed handheld PCR technology so that we could do this on site. And in fact, we have done a project like that with our fourth-year biodiversity capstone course, working with Trout Unlimited and the City of Guelph to do on-site monitoring for brook trout right here in Hanlon Creek. So we can do it on site, but what we've learned is that your power to detect something grows with the more samples you take. So looking for eDNA isn't just a test, it's an experimental design. And so if you really want to have a high probability of detection, you need to take multiple samples. And so at that point, it becomes more scalable to run them in a lab than to do repeated tests with a handheld instrument. Yeah. But in some cases, that still has its utility.
For sure. So we aren't exactly at tricorder level detection yet, and still relying on that laboratory-based testing, which takes me to effectively the innovation that you have. Conceptually, what you just described, you were able to port that to a commercial environment with a company, Precision Biomonitoring. Right. Amanda Baum, who you've already mentioned, a postdoc in your lab, formerly a PhD student,
yes,
and then went on to do a postdoc. Yeah. And together with someone who operates more in that commercial space, Dr. Mario Thomas. Mm-hmm. Mario and Amanda, co-recipients of the Innovation of the Year Award with you. Yeah. Tell me a little bit about that particular journey and taking the concept and mobilizing it into the commercial space.
Again, great question. So we see the utility of developing these kinds of assays for species at risk, initially here in Ontario and also more broadly. But to take an innovation out of the lab and into a commercial setting requires a significant amount of R&D. And so in this case, what really catalyzed this shift was an INSERC Idea to Innovation grant that is designed specifically to do that. And then we were fortunate enough to match that with provincial funding as well, so that we had support to develop more of these assays that could be commercialized with this new entity, Precision Biomonitoring. So it really began with financial support from the provincial and federal governments.
That's fantastic, and it speaks a lot to the great work that is done by our provincial and federal governments to identify projects to provide this sort of support to move them into the commercialization space. And boy, that's something that you and others have really taken advantage of here at University of Guelph, and it's fantastic. Any places that really stand out with you that say, wow, there's an instance where this innovation has really made a difference?
Yeah. I would argue that here at the University of Guelph, our motto is to improve life, but to improve human lives also means to sustain biodiversity and the ecosystem services it provides that are crucial to our own survival. So for some companies, they may not have liked the answer that, say, red-side dace was in an area where they wanted to propose a development, and detecting that species and protecting its critical habitat may not have been something they were happy about. But in other instances, when we've worked with non-governmental organizations like Trout Unlimited, who are interested in knowing which remnant habitats still support brook trout, We did this project that I mentioned with brook trout here in Hamlin Creek, paired with electrofishing, working together with some local environmental consulting firms like NRSI and SLR, where their people came out, did electrofishing, our students did the eDNA testing, so we essentially validated that the method worked. We found fish with electrofishing, we found it with eDNA, so then we could move to spaces where we didn't know whether or not brook trout still occurred. So in this case, Trout Unlimited was really keen to know, are there still brook trout in some of the headwaters on the Niagara Escarpment? It's private land, it's not easy to access to do these kinds of surveys, so nobody had looked in decades, but we could just stop at the side of a road, take a water sample at a culvert that, you know, where some of these streams ran through, and we showed that on 12 Mile Creek and other places here in the escarpment, there are still brook trout populations. That now Trout Unlimited is excited about trying to develop community programs to protect that habitat.
Nice, absolutely excellent. I liked your first response where some of I like the entirety of your response, but I also like the first part where it said, you know, the company may not have been happy to make this discovery that there is a at-risk species on the site where they had wanted to develop, but they've now been able to make a better decision. They've been able to not just be a greenwashing company, but one that's actually being adherent to their fundamental principles, and that improves life arguably for them as a company as well. So,
I would agree. In the long term.
In the long term, yeah. No, everything about that answer was absolutely fantastic. Now, a stereotypical question that we ask about that journey that you've just described from fundamental concept through to something that's realized in a commercial space and that's now making an impact in everyday people's lives that enables the University of Guelph to fulfill its mission to improve life. You've gone along that pathway. I've heard you say in the past, I don't know if I would encourage someone to go that innovation route, but clearly you've been successful in that particular handoff. It's clearly involved partnership. Is that the advice that you'd give people, that you know, get the partnership right to make things happen?
Yeah, absolutely. And I would say it's partnerships. If you're an academic researcher, you know, you're not going to commercialize something by yourself. Or if you are, you're going to have to leave your academic position. Right. You know, you can't have it both ways. Yeah. If you want to see some of these innovations commercialized, you need to build that team. So, you know, crucially, with the support from INSERC and the province through MyTax, I was able to recruit highly qualified personnel, PhD students, postdocs, to continue to develop the innovation, also to work with a startup company, because that's the other piece of the continuum. As a researcher, most of us don't have the expertise to raise capital. And that's another piece of the partnership. You need a management team that understands how to commercialize, and then you need early investors. And Precision would not have been able to get off the ground without that total package of a management team, early angel investors to take it to the next level. So it requires a multitude of partnerships, not the least of which with the University of Guelph, which, you know, I'm fortunate enough to be in an environment that fosters commercialization.
I could not ask for a better answer, Bob. Really appreciate the feedback and the insights there. And I just want to finish by saying, again, congratulations to you and your team. I think that's really, really an important message to you and your team for being recipients of this year's Innovation of the Year Award. One of our 2 recipients this year. Absolutely amazing work that truly is making a difference. And as both of us said earlier, is enabling University of Guelph to realize its ambition to improve life. Thanks so much, Bob.
Thank you.